Right, I'm Malcolm Bennett. I'm a professor at the University of Nottingham, and I'm currently a BBSSE professorial fellow. So that gives me 5 years of no teaching, which is a real luxury, and it's a privilege.
Can you describe the area of plant science you work in and explain some of the work that's currently happening in your lab?
Yeah, I work on what people have described as the hidden half of plant biology. So I work on root development. And for many years it's been the kind of area that's been quite neglected. But with the use of plants like Arabidopsis, we've been able to identify a lot more, or discover a lot more knowledge about how the genes that control root growth and development. And recently my lab has been trying to get out of the Petri dish, where we tend to grow our plants and look at the roots, and into the soil. So we're working with soil scientists like Sasha Mooney and computer scientists like Tony Pridmore at Nottingham, who've developed really neat techniques to actually visualise roots growing in soil. So it's really taken our work into another direction. So we're starting to really study root development in their true natural setting.
If that sort of relates to my next question, is there a link between what you do and sort of everyday life or practical aspects like that currently?
Absolutely. We hear a lot about global food security and the importance of that. And since the Second World War, the food surplus we've obtained, the increases in yields have been largely down to the Green Revolution. And this is where people have got dwarf plants, and they'd actually, so that a lot of the energy goes into the ear rather than the actual stem. And this has been an absolute triumph. But more recently, the yields have tailed off, and these varieties probably haven't been selected for their root architecture traits. Now, roots are very, very important for things like the uptake of nutrients, drought resistance, and so what we aim to do is to actually try and re-engineer root systems to include these beneficial traits. And the whole idea behind this is we'll make them more resistant to climate change, okay, and also more sustainable, because they'll take up more nutrients with less addition of nutrients, and so they'll take them up a lot more efficiently. That's the concept. So essentially we want to re-engineer root systems architecture.
Excellent. What do you think the major challenges are for plant sciences in the next 10 years?
Well, we've certainly got the public ear. Global food security has really captured the public's imagination. So I think that's fantastic. We have more public support. I think funding is going to be a tremendous impediment. Obviously, there have been serious budget cuts across the public sector, and so that's obviously going to limit activity within the UK. But if you look at the kind of investments going on in places like China, there's a 20 to 30% increase year on. So there really is a disparity between different countries in investment in plant science. It's sad that Britain isn't really capitalising on the investment it's made over the last 30 years and the lead it's had. I am concerned that we will fall behind as a international power in plant science. There's a recognition in the research funding councils of the importance of this, but whether it translates into more funding is another question.
So how do you think the plant science community in the UK should structure itself to meet the challenges you've just sort of mentioned there? Well,
clearly we need to be more effective at lobbying the government, and so there's been some really good ideas and structures put in place. So the Plant Science Federation, I think, is a really great development. This provides a really effective body that represents not just Arabidopsis but also crops, horticultural crops, arable crops. The community, the research community that work within those, and the industries as well. So this is a really, I think, going to be an effective lobby group. And also the creation of the Society of Biology. That's really one voice. Finally, we have one voice in the UK the government can speak to. And I think over time these organisations are going to be extremely effective.
Given that new technologies are lowering the barriers to studying agriculturally and commercially important crops, what role, if any, do you think the model plant Arabidopsis has to play in the future of plant science?
I mean, I've always described Arabidopsis as a genetic treasure trove. It's so much easier to work in Arabidopsis for lots of practical reasons. It's a higher plant with almost identical genes to many important crops, and so I think, and we know from our own work, that we can study genes in Arabidopsis and study the equivalent gene in rice, and they have conserved functions. So, you know, the fact that these genes are divided by 100 million years of evolution, and yet they retain gene function means that Arabidopsis is still highly relevant to studying such highly conserved functions. I mean, clearly some genes functions are going to differ between different plant species, particularly genes which evolve at a rapid rate, like disease resistance genes. But genes which are highly conserved are likely to be easier to study in Arabidopsis. So I think it's still highly relevant. But we have to seriously think about translating our discoveries into crops. So we've had 20 years of great investment in developing Arabidopsis, but we've really got to make it relevant to today's challenges, agronomic challenges.
And what do you think the barriers to that are?
I think a lot of Arabidopsis scientists are reluctant to move into other systems. I mean, clearly they're not going to have the same resources that they've been used to, but they're improving all the time. I mean, there have been tremendous developments in rice, for example, and also wheat genetic resources. The UK government's put a huge amount of money into, for example, the wheat pre-brooding work, which is going to generate some fabulous new germplasm, which I think Arabidopsis researchers could really exploit to study their traits of interest. So I think that the barriers are almost intellectual rather than and practical, but we have to rise to that challenge.
Excellent. In your recent work, you've utilised systems biology approaches to further understand key questions in biology. Do you think that this type of approach is generally applicable to all areas of plant science?
I think that it's really helped my biology to go from almost qualitative, purely genetical, to far more quantitative, realistic understanding of the systems I study. So that's extremely important as we move from studying individual genes and their gross effects, say in an Arabidopsis mutant, to understanding the roles of these different genes within the context of a network of genes. We have to understand their quantitative contribution and the contribution of different signals. And many of the pathways we're studying are very complex, and so the outputs of these pathways are non-intuitive or non-linear, and so we have to use mathematical modelling to be able to simulate the outputs, and which we would test experimentally. So maths has had a huge impact on me. Imaging and computer science has also had a tremendous impact, far more than I anticipated, in terms of extracting information from images. And this isn't just relevant to Arabidopsis, but also to crops. We're using really advanced imaging techniques to study crop root architecture or crop canopy structure. So computer science is really a very important role. Engineering is also increasingly important discipline in terms of understanding the biomechanics that underpin plant growth and development. So yeah, I would say that the systems approach, which really embraces maths, computer science and engineering out with biology, they have huge contributions to make.
You've touched on it before, but these technological advances, are they going to help you to take the model plant into agriculturally important crops and plants? Specifically on that.
That's a good point. So coming back, I'll give you one very simple example. We want to visualize roots in soil. And so we've been working with the computer scientists to develop tracking techniques which are used to, for example, track humans in browse. And we've used the same concepts to actually track objects through X-ray slices to actually then reconstruct root architecture of crops. So these are tools which we're adopting from human research or Arabidopsis research and applying to crops. For example, we're also using other techniques. I described one in the lecture today, tracking root angle. Root angle is very important for lots of different traits such as nutrient uptake and water uptake. And we were using a technique which is currently being used to track missiles at the angle of missile noses. And so, we take a lot of technology and apply it to plant and crop science. I think that's how we should be.